Hey everybody. Alright, so we're on to our last topic, which is for us to pretty extensively step through this paper from Genentech on a Tubulysin antibody drug conjugate that enables activity against multi-drug resistant tumors. It's a pretty cool paper. Little complex here and there, but we'll do our best to step through it, and I'll try to go slow on anything that's a little complicated for you. You may have to watch this twice to grasp it, but anyway, let's get started. I'm going to actually go back to the last slide, which was on the total synthesis of Tubulysin B, just to remind you what the structure of those things is. So let's see. Yeah, this is from the last lecture, Tubulysin B, which is in the whole class of these tubulysin molecules. They're kind of peptide-like, and we showed the synthesis of it. What's Tubulysin B versus epi-Tubulysin B? The epi-Tubulysin B just has a different chiral center on the C terminus right here. A natural CH3 versus non-natural CH3, and that didn't really have an effect on the activity versus the synthesized tubulysin B versus the synthesized non-natural Tubulysin B had basically the same potency. So that was interesting. Okay, let's move on to the new paper, which is an antibody drug conjugate based on Tubulysin. So this is the paper. The reference is the paper is actually on the iLearn site, so you can download it from there. And with a lot of papers, this has a first author. So this person, I don't know what their contribution was, but usually it's kind of like their project. But then you also have like the last author, who is maybe runs the research lab or something, maybe a principal investigator. And that's Thomas Pillow. And Thomas Pillow's done a couple pretty amazing papers in the last several years on antibody drug conjugate things. So you might see his name around if you look around in the ADC literature. And they're at Genentech, and Genentech's one of the leading companies for antibodies and antibody drug conjugates. So this is a pretty cool paper. Abstract, I'm not going to step through it because we'll see the paper shortly, but this is the main antibody drug conjugate that they worked with, and the abstract is essentially that they found acetate at the R position, acetate, which is C double bond O CH3, to be unstable. It had some in vitro activity, but then they put it into a mouse, and it did not survive, and the compound got destroyed. And anyway, so what they basically this whole paper in a nutshell, and that's what the abstract does, is that they turned it into a propyl ether. So it's oxygen, carbon, 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 and that made it stable in vivo and good efficacy. So ton of work. It's a ton of work, and it's a cool paper. It's a cool story. So let's get going on this cool story. So a lot of this I'm just going to kind of read from the paper and maybe comment on as we go along. So this is from their introduction. Over the last 40 years, antibody drug conjugates have been employed to rescue highly potent cytotoxic small molecules that exhibit limited therapeutic activity due to dose-limiting toxicity when administered as free drugs. That's a powerful statement. And what it's saying is we have a lot of compounds that are very cytotoxic, and they will absolutely kill cells. They'll kill the worst of cancer cells. But the problem is they are toxic. If you inject a lot of these drugs, like Taxol, depending on what concentration you use, they're very, very poisonous, vinblastine, etc. So there's the issue is toxicity, and how can we avoid the toxicity of these substances? The way to do it, one way to do it, is make an antibody drug conjugate. So you're attaching the drug somehow to an antibody in a way that it links to it, and it can be cleaved, and and then kill a tumor cell, for example. Okay, so tubulin inhibitors, which we talked about last time, in particular have comprised the vast majority of drug types utilized in ADC clinical trials. So we talked about a bunch of them. We talked about Taxol and vinblastine and vincristine and all those various things, the vinca alkaloids. Tubulysin, and we saw the synthesis of a tubulysin analog last time. It is also a tubulin inhibitor, and almost all of the tubulin inhibitors in antibody drug conjugates are sort of in the auristatin class, and also the maytansinoid class, and these are found in clinically approved antibody drug conjugates Adcetris and Kadcyla. I might be mispronouncing that. And so this is monomethyl auristatin. We did see this in the last lecture also. It's one of these things that people have been attaching to antibody drug conjugates as a cytotoxic peptide. And also mertansine, which is one of these maytansinoids. And this is also another thing that binds to tubulin and is cytotoxic. And both of these are called second generation ADC warheads. Okay? And what we'll see, Tubulysin is considered a third generation one. It's the one that's the topic of this paper. Also, just kind of memorize MMAE or remember it, because we're going to see it about 1,000 times in this presentation. And it's kind of referring to, we'll say, almost like a competing ADC warhead. Okay, so everything that they do in this paper is going to be kind of in comparison to MMAE. Okay, so keep that in your mind. All right. So the tubulysins, which we've learned a little bit about now, similar to the auristatins in their linear peptidic structure, are a class of highly potent cytotoxic natural products. They inhibit tubulin polymerization and disintegrate microtubules in dividing cells. That's kind of cool. So if you can get this stuff into a cancer cell, it'll kill it. It'll be cytotoxic, and it'll destroy tubulin, and it'll inhibit those little polymeric networks of tubulin that are used in the mitotic spindle and mitosis, right? That's kind of cool. Tubulysins bind to the vinca domain, vinblastine, which we showed that last time also. We showed the structure of tubulin and where the vinca domain is. It's just a binding pocket on tubulin. But unlike monomethyl auristatin E, these are able to bind to the beta subunit alone, and they we also showed that. There's an alpha and beta subunit of tubulin. And these bind to, maybe with a high affinity to the B subunit of tubulin. The tubulysins do. Okay, and then this is MMAE, and which we've seen before. It's part of this Adcetris, clinically approved antibody drug conjugate. And then this is NH tubulysin M, and we'll explain the NH. There's several of these, as we mentioned. The one we showed the total synthesis of last time was tubulysin B, so it's a little different also. In general, they have the same general structure. Okay, but this NH we'll see is kind of interesting, and it will be a little bit of an issue in the usefulness of this tubulysin. So keep an eye on the NH. Okay, and so, but overall, we could consider the MMAE like a second generation antibody drug conjugate warhead, and the right one, these tubulysins, NH or others that we'll see, as a third generation warhead. One of the most compelling reasons to investigate tubulysins is that they are not effluxed by PGP11, unlike MMAE, which is. This is a big deal. You may have remembered PGP because we did talk about it earlier in the course. is P-glycoprotein. It's a transport protein used by multi-drug resistant cells, and there's also some various tissues that try to efflux out drugs that they might consider toxic. Weird that tumor cells actually try to efflux out anti-tumor drugs. And what they're saying here is that tubulysins are kind of resistant to that. That is a big deal if you're trying to treat multi-drug resistant tumors. So we're interested in exploring the impact of chemical diversity, increased tubulin binding, and lack of efflux on the efficacy and safety of a tubulysin antidrug conjugate compared to an MMAE antidrug conjugate. So they're going to basically they're going to look at ADCs, antibody drug conjugates, for either tubulysin or MMAE, and then they compare them, and they said, oh, well, the latter one is pretty well characterized preclinically and clinically. So this is actually in the clinic, and they're saying, well, this is maybe better. That's kind of what they're trying to say here. And you can envision that there's a little bit of a financial interest here. This, I believe, and I don't follow all of the financial issues with drug companies. I think this was designed by a competitor drug company, Seattle Genetics, and I may be right, I may be wrong on that. Whereas I think Genentech's kind of pursuing this other one. Okay, so what does PGP do? We showed this figure before. I'm not going to really go through it again, but this just kind of shows all the various things that P-glycoprotein does, and it exports drugs from various organs or effluxes them, so it kicks them out, and including your liver and your intestine. The key one here is tumor cells. Also the brain. I think we saw it earlier with the brain. So it's a efflux system that kicks stuff out of your central nervous system. Anyway, so yes, tumor cells use this also. Little arrows kind of show how drugs getting kicked out of tumor cells. And this was also a X-ray crystal structure we showed previously from Nature. I'm going to try something. I want to see if I can move my face up. There we go. Okay, so if ever I'm covering something up at the bottom right, I can move my face out of the way with my program I'm using. But yeah, Nature 2012, page 566 to 569, and this was showing the X-ray crystal structure of what the PGP glycoprotein looks like. So it's a well-characterized protein. I think this is from a ringworm or some other species. So I don't know if they have a structure of it from human, but it gives you an idea what it looks like. Okay, let's see if I can move me back down here. All right. Okay, so tubulysin antibody drug conjugates are based on a carbamate linkage. So this is how they kind of get connected to the drug. And so this is a long string of text, MCVC PABC linker. It's just a linker they use to attach the antibody to the drug. It's utilized in many clinical ADCs. It's highly stable when attached to specific sites on an antibody, and it releases primary or secondary amines when cleaved by lysosomal proteases, cathepsins. So cathepsins are going to be it's a protease. It cleaves peptides, and it's in the lysosome. And it turns out when these antibodies get imported into a tumor cell, so they get captured. It's kind of like the antibody connects to the cell, and then it gets sucked into the cell, and it makes something called a lysosome, which is a little pocket with a bunch of protease enzymes and things. It gets destroyed. It dissolves everything. It dissolves the antibody, destroys it. And these proteases also include cathepsins, and guess what the cathepsins do? They release the drug. So the drug gets into the cell, and now it's cleaved off, and it's a free drug, and it gets to do all its nasty drug stuff, cytotoxic stuff. Okay, So this is kind of one example of how this might look. So the MC, VC, PABC is kind of the whole left side of this. And the right side is tubulysin. This is tubulysin connected via a carbamate linkage. It's the little linkage right there. And what happens is that cathepsin, the cathepsin proteases, cleave the C double bond NH linkage right there. So it cleaves it. It pretends it's a protease or a protein or peptide bond. It's not, though. But anyway, cathepsins, this is one of the things they do. They cleave the bond. And when that cleaves, now you have an NH2, a benzene, and this whole thing. And this, I think they have a name for it. I want to say self-immolative linker. Self-immolative linker. And it basically what it means is once this happens, when this breaks off, this falls apart also. This little linker falls apart and kicks out CO2. So CO2 gets lost, and you get your amine. You get a secondary amine. And this can work for primary or secondary amines, okay? And then they've released the NH tubulysin M. And the NH basically means secondary amine right here, okay? We'll see that that's going to be a problem in a second. Okay, what does MAB stand for? It's monoclonal antibody. So it's an antibody that has a specific epitope. There's a lot of immunology about antibodies. I'm not going to get into it, but it's just one of the kind of really specific types of antibodies. It's kind of a biology question, but yeah, it's just a protein that is very specific for markers, in this case, on tumor cells. Okay, and also we have a secondary amine. This is producing a secondary amine. Okay. One could envision using this strategy to generate a demethylated tubulysin analog, secondary amine, NH-tubulysin 1. So it's like this structure right here. Yeah, you could totally do this. You make this, and then you pretty much give it to an animal or something, or a human, and it will find its target through the antibody, get sucked into the tumor cell, into the lysosome. Cathepsin will eat it up. We'll just chop this bond. Now this all falls apart. You have your cytotoxic substance. Sounds good, right? But tubulysin M, which is the version with a methyl group, so tubulysin M is a tertiary amine, not a secondary amine. It may, and this is known to be cytotoxic. So this would be ideal, right? If you could somehow have tubulysin M. And the same kind of thing, but just imagine a methyl group, and then you'd have to have a methyl group out here, right? But would that work? Because it's like now it's like a positively charged carbamate. That seems a little weird. We'll see that it does work. But that was a worry on the outset of this paper. Okay, so this would be the tubulysin M molecule. It's a tertiary amine, right? And it's not the same as a secondary amine. And the only difference is you need this whole molecule, but with a methyl out here first, and it'd be a positively charged methyl. Okay. All right. And that'd be a tertiary amine. So we employed this strategy with the easily accessible, potent tubulysin M analog, a potent analog. And that would generated the NH version, and it's carbamate linked ADC using, and they attached this to an antibody that targets tumor cells. It's called a CD22 targeting antibody, and they engineered a cysteine. So they attached basically what they did is they took the antibody, which had a lysine at position 149. And then they mutated it with site-directed mutagenesis, which is just a molecular biology trick. And that converted lysine 149 to cysteine 149. Okay? And so with this, what they basically did is they have the linker and they have the tubulysin without the methyl. And they attached it to basically to cysteine 149 that they engineered. So it's a Genentech, one of their tricks is they like to take antibodies and modify them and attach things to them. So now they have this specifically at a specific amino acid on the antibody. Okay. All right, and then they would, like we said, envision cathepsin, chop the bond, fall apart, and now you have NH-tubulysin M, okay, versus the N-methyl one. So I basically showed you these structures already. Secondary amine, tertiary amine. Unfortunately, consistent with other literature, removal of the methyl group to enable linkage to the antibody-drug conjugate resulted in a dramatic loss in potency. Isn't that crazy? Because we talked about how, like, in Tubulysin B in that last paper, last lecture, how we just changed the stereochemistry on like the C-terminus and it had no effect at all. But on this, it's like if you get rid of a methyl, the methyl is really important and it demolishes activity if you get rid of that methyl. One little atom. Okay, so the NH one's really bad, and it's much less potent than Tubulysin M. Okay, so keep that in the back of your mind. N-methyl good, NH bad. Also, the other thing they did is, so this is compound 1, which is NH-tubulysin M. They also looked at the antibody-drug conjugate, which is, they're calling compound number 4. This is connected to the antibody. It's CD22 is anti-CD22. CD22 is a marker on tumor cells, on lymphoma cells, and so they have this antibody. It's anti-CD22, and they also, as they show here, they looked at this antibody-drug conjugate and found it inactive against lymphoma cells too. So not only is this bad, this is bad also. 4 is bad, 1 is bad. 2 is still good. Okay, so alternatively, they're like thinking, is there a way we can modify the ADC structure to allow you to have that methyl group? And it says, they say, alternatively, it generated a different ammonium-based ADC using a protecting group-free synthesis. So this is organic synthesis. It's pretty easy chemistry, though. We generated in just 3 steps from a commercially available starting materials. This shows the top one that I already showed you with the NH. This shows the bottom one. So what they're doing now is they're actually having the methyl group in the ADC. And now it's a positively charged nitrogen. And as an organic chemist, I always get scared of molecules that have a positive charge in them like this and built into an organic molecule. But somehow this worked, and it wasn't that bad. And they're calling it compound 5. I'll talk about 6 and 7 in a bit. But when compound 5 is clipped by a cathepsin, it cleaves the bond, now this whole thing falls apart, and now they have the key N-methyl that they need, and now it's Tubulysin M, which is actually potent, and we'll see that in a second. So this is really cool. Okay, so they went from a secondary amine, and then here's a quaternary ammonium, which just means the actually, that's interesting. Let me see something real quick. Oh no, they did change the structure a little bit. Yeah, this is a carbamate. Yeah. I just realized something. Yeah, this is a carbamate linkage right here. This is a quaternary ammonium linkage. So they're getting rid of the C double bond O and the oxygen. So in the end, though, it works the same. It clips, this falls apart, this cleaves off, and then they're left with Tubulysin M, the good cytotoxic compound. Okay? So now it's a tertiary amine. Okay. So the resulting anti-CD22 Tubulysin antibody-drug conjugate, compound 5, which is, this whole thing is compound 5, was potent in vitro, and it's at this point, it's about the same activity as MMAE ADC 8, which was the other one, the auristatin one, the competing company one, against lymphoma cell lines. And lymphoma, again, is a cancer of lymph node cells, and kind of like they're related to immune response, and there's a lot of biology behind them. For now, we're just considering them just one of it's a cancer drug target. Okay. The other thing, just to mention on the outset, ADC 6. What is ADC 6? So 6 is the same exact thing. It's a negative control, The big thing is NAPI, this thing NAPI. It's a different antibody. And it's one that does not, it targets a different antigen, a different protein, like on the cell. That's not CD22. CD22 is like a target for an antibody for lymphoma. Okay, so ADC6 is a negative control because it's not targeting the same target antigen, CD22. Okay, and the other one that's kind of cool is another control, number 7. So what 7 is it's uncleavable. 7, well, we'll see this structure in a second. The big difference is that in the citrulline, which is like next to, it's a part of the where the cathepsin recognizes the protease that cleaves the bond. It is very picky about stereochemistry, and ADC5 has the natural amino acid stereochemistry. ADC7 has the opposite stereochemistry. And so cathepsin doesn't know what to do with it. Cathepsin can't cleave it. So I say ADC7 is uncleavable. Okay? And both of these are negative. They don't have activity. So these are just really good, well-designed negative controls in their study. Okay. All right. So in vitro cytotoxicity of tubulysin and the ADCs. Okay, so there's a lot of information here, and I'm going to step through it very slowly and carefully. Okay, so tons of numbers. Okay, so I'm going to go through each key conclusion. First one, unconjugated tubulysin M and MMAE. So tubulysin M is the one with the methyl group. Good. MMAE is the competitor peptide. So both of these are actually potent, and that's what we're showing here. So BJAB and WSU are lymphoma cell lines. Jurkat cells are an interesting one. It's a different cell type, but they don't have that CD22 thing on it. Okay, and so it's a different tumor cell, but it does not have CD22. Whereas BJAB and WSU, these are lymphoma cell lines, and they do have the CD22. Well, interestingly, well, these compounds, though, are cytotoxic, right? They're both cytotoxic. And so they're just killing tumor cells like crazy, right? And they don't, even though Jurkat doesn't have CD22, it doesn't matter because these don't have an antibody on them, right? They're just the peptides. They're just cytotoxic peptides, right? Interestingly, also, though, this second column has PGP. It's overexpressed with PGP, which is the efflux protein. And you can see this is a really important thing. MMAE, the competitor drug, is really terrible. So it's very non-potent against the PGP version. Okay? Okay. Then we say NH-tubulysin M, which is the one without the methyl group that we saw. This is less potent. And it is. It's not as good. It's not as good as tubulysin M. So that methyl group on tubulysin M is good. Okay? And also, like I said in the last slide, the antibody drug conjugate with CD22 specificity of the NH version of tubulysin, right? The NH tubulysin one is totally dead. And we just said this in the last slide. Yeah, that's that one. It's really big numbers, really bad, right? So far the conclusions are compound 1 and compound 4 are kind of bad, and compound 4 is very bad. Okay. What else? Tubulysin-based antibody. So tubulysin-based antibody drug conjugate number 5 and the MMAE-based antibody drug conjugate number 8, those were potent against BJAB and WSU, lymphoma cell lines. So essentially we're taking compounds 2 and 3 and we're making antibody drug conjugates now with the antibody attached. Okay? And we see 2 makes 5, 3 makes 8. Okay? And number 5, and they're both pretty good. Right now, against BJAB, lymphoma cancer cell line, and WSU, those are pretty good. Those are pretty good, yeah? Significantly, the new ADC5, the new one, number 5, retained activity against the PGP overexpressing cell line, while the MMAE one did not. It's completely dead. Look at that. So it has activity. It's not great, but it has some activity. Whereas the MMAE one, the competitor cytotoxic peptide, is terrible against PGP expressing cell lines. And antibody drug conjugate activity was antigen specific. So we're talking about the Jurkat cells, right? And those do not have CD22. It's just a different kind of cancer cell that does not express this protein on its surface. And so that's kind of a good result. It shows that this compound 5 actually is targeting CD22, which is a surface protein on lymphoma cells. And of course, this one is also terrible against that. That totally makes sense. This makes sense based on the fact that the antibody is CD22, and you're looking at a cell type that does not have CD22 on it. And then the other control is that what about doing the same, using the same good Tubulysin AM, but rather than have an anti-CD22 antibody on there, how about having this NAPI antibody, which is just a different protein, it's a different antibody that does not target CD22. Guess what? It's dead. So that's also a good sign. It's showing that you need that anti-CD22 antibody, right, to attack these lymphoma cells. Lastly, they see inactivity of Tubulysin ADC 7, which, like I said, I just showed this on the last slide, is the opposite chirality at citrulline. It's what's needed for the cathepsin cleavage. So that's this, and that's just showing total dead activity. Compound 7, completely dead. And this shows what that looks like, compound 7. So it's citrulline is this weird amino acid out here, and this is the non-natural stereochemistry. And it's resistant to cathepsin. And guess what happens when you make an antibody drug conjugate with your cytotoxic peptide, and it can't be cleaved by cathepsin while you have an inactive compound. So you could say that 6 and 7 are really good negative controls. It's like, to kind of make sure their assay's correct. Okay. And one could say that compound 5 looks pretty good, right? That might be a conclusion so far from this table. Like if I was putting some money on the table, I'd say, oh yeah, let's spend that money to develop compound 5, right? Right. Okay, so in vivo efficacy and stability of this kind of, we're calling it first generation Tubulysin ADC 5, which is the one they just created, compared to MMAE, which is the competitor one. In vivo efficacy and stability, this is like, does it work in a living organism, and is it stable? So they selected this ADC5, and they evaluated in a human lymphoma xenograft. So all that means is they took human lymph node, or I think lymph node cancer cells, and they put them into a mouse, and then they're able to kind of watch the tumor growth and see if the drug has an effect. Okay, so here's number 5, the money one so far. Okay, it's got the ammonium linkage, yeah. Okay, and the bad result. Unexpectedly, ADC5 showed very modest tumor growth inhibition, TGI, almost like, I don't know, think of TGIF, it's almost TGIF for us. TGI, after a single IV dose, 1 mg per kg, which is not very much compound, right? Whereas the competing one actually kind of worked. Uh-oh, that sounds kind of scary, through day 28. So what this is kind of showing is that in this kind of simple tumor model, the competitor drug's working pretty good at the same dose, right? And it's red one's killing tumor volume, right? Whereas if you black vehicle basically means no drug at all. Well, the tumor's just going crazy, growing like crazy. And then the blue compound, compound 5, is kind of not really working too well, right? So that's kind of scary, and that's for the commercial development of this compound, that might be a bad thing, right? So this in vitro in vivo disconnect led us to investigate the in vivo stability of the two ADCs, 5 and 8. And what's 8? 8 was the 8 was the MMAE one, right? 5 was the one they were creating, right? Okay, so just like, let's check the stability of these compounds. They so both ADCs are comprised maleimids. That's just how they link it to the antibody, conjugated to the same site. So the same site of the antibody, LCK149C. What does that mean? LC means the light chain. It's just a antibody nomenclature. It's one of the pieces of the antibody. K149C means it's lysine 149 gets mutated to cysteine, so they do molecular biology. And they found this drug to antibody ratio of 2. So they so that's kind of like they attached the drug, and then there's 2 drugs per antibody, right? Drug antibody ratio. That antibody site was selected because it forms highly stable connection to maleimid containing linker drugs. I mentioned this before because Genentech is really good at finding good places to put linkage sites. It's like one of I think they have a whole division of the company that does that. Okay, so then they did LCMS analysis, which is just HPLC mass spec technique, and determined in vivo stability. And what they noticed was the acetate was getting chopped off. So, we talked about metabolism in this course, how things like esters can be cleaved. It's probably maybe an esterase or something, just clipping off the ester, making the hydroxy group. And then this is totally dead, it looks like. They measured the drug activity ratio for 5 to 8 over time, and look at this kind of sad result. It looks like compound 8 is totally stable, the MMAE one, the competitor one, and compound 5 is just being destroyed and hydrolyzed. Okay, so you could also say, that's kind of bad for the commercial development of compound 5, right? So then they're thinking, what can we do? What can we do? They explored 2 approaches to improve the stability. Okay, I think there's a lot of text here, and I'll go through them though. Method 1 basically means find a different place on the antibody. Rather than that cysteine location, they can use something called cysteine engineering to identify an antibody site to protect the conjugated small molecule from metabolism. So basically just link it somewhere else in the antibody and do this very rationally. And they did this, and they actually wrote a second paper. We're not going to talk about the second paper. It's actually it says in preparation, but it's actually prepared and done and it's published. If you want the reference, let me know. I can send it to you. Method 2, a more general solution, not requiring antibody engineering, is let's just try to cleave the acetate with something else. And they're thinking an isoster basically means like something that has the same kind of binding properties. And tubulysin analogs with propyl ether or propyl amide also afford low nanomolar potency. So basically what they ended up doing is they just envisioned taking off the acetate and having a propyl group there instead. Okay, and ethers are, they cleave, but maybe with P450 or something, but much maybe a lot more difficultly than an acetate. Ester is just a very cleavable thing, and that makes something called tubulysin PR, compound 9. Remember the the tubulysin M was compound 2, and the one with the NH that we saw earlier, what was it over here? The NH one was tubulysin, NH tubulysin, and that was compound 1. So compound 1, 2, and now we're on to 9. Okay. Okay, so it looks like maybe the acetate's bad, and the propyl, yeah, it might be good. And I think they had some evidence that said that it actually is good before they started going down this route. Cool synthesis. This is a. I'm not gonna really spend any time on this synthesis except a couple steps. This is in the supporting information, and you can step through this yourself, I guess. They did a couple steps to build this chiral intermediate number 8, including this sulfur thing kind of set the chirality. So this is called an antioselective chemistry, where they use one chiral thing to induce chirality on another thing, and then they cleave it off, and then they're like at this point, and then they do reductive amination, so organic 2, right? Nitrogen makes an amine, and then they reduce it, and that's how they attach the propyl group. Yeah, this is maybe, I think, one of the coolest steps, which is how they built this amide linkage. Well, this is not that exciting. This is just like a amine reacting with an acid chloride, right? So good leaving group kicks it off. But the next step is, and I think we saw this in that other Tubulysin paper. They have an azide, and then since they're building a very congested area of the peptide, the azide trick seems to work pretty well for that, and I think the trick was when this bond is formed. If you have a protecting group, like a normal nitrogen protecting group, that there's problems, and it doesn't give you a good yield. And then you can just consider the azide as kind of like a protecting group, right? Because once you attach the nitrogen to this and make the amide, then you have the azide, but then you can cleave off the azide and make an NH2. So this is you can kind of consider azide as a protecting group, right? Okay, then they just do some more stuff, kind of standard peptide coupling to the this nitrogen thing, and then lithium hydroxide cleaves the ester to a carboxylic acid. They use DIC. Remember DIC? It's another carbodiamide coupling agent. Pentafluorophenol. Saw that in the last paper, and essentially then that forms this bond. Yes, makes the amide linkage right there. Then they attach this is Tubulysin PR. This is the cytotoxic drug. And then this is kind of basically the whole linker thing with the valine, citrulline, and then the maleimide. This is kind of cool. This is what we connect the antibody to, right? The antibody has a cysteine nucleophile. This is an electrophile. So anti-CD22 cysteine engineered antibody, and we call it the LCK149C light chain, and this is the cysteine residue, and that's how the sulfur of the antibody is a nucleophile. It connects to the maleimide. So this is a definite nice connection between a biological idea, an antibody, to an organic chemical idea. This makes the antibody drug conjugate. Very cool. And then this is just that, focusing on that step again, the azide step. The idea that if you have you want to form a bond between the nitrogen and like this carbon, and the Fmoc is a protecting group. It's a common protecting group in peptide synthesis. I don't know if we talked about it in this class. Anyway, it's just a protecting group, and 5% yield, terrible, 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 right? And if they swap the Fmoc amino acid chloride for the azido acid chloride, they get a 14-fold increase in yield in the desired amide, and then reduce the azide, and you get to the same place. So now it's like beautiful, right? So the nitrogen kicks off the chloride, it just makes an amide bond, and then you reduce the azide to the amine. So great. This is a great example of a synthetic kind of optimization. All right, so this is the table one that we sort of populated before, and this is kind of showing that compound 5 was kind of our good one, remember? And compound compound 5 was kind of the good antibody drug conjugate that then turned out to be bad. with in vivo testing. So with improved tubulysin, the propyl ether one, analog 9, we were able to remove the metabolic liability, right? So it doesn't metabolize, while retaining the low double-digit picomolar potency found in tubulysin M, compound 2. That's really cool. It's really important. So that changing the acetate to the ether did not destroy activity. So if we look at compound 2 and 9, yeah, they're sub-nanomolar, right? It's a decimal of a nanomolar. 2 looks really good, and a compound 9 looks really good. Yeah, they're, maybe not perfectly perfect, but good, sub-nanomolar, right? So that's good. And then the retained potency was also observed in the antibody-drug conjugates. So now they take the peptide, tubulysin propyl ether, and then they make a antibody-drug conjugate, anti-CD22 antibody-drug conjugate, tubulysin propyl ether, compound number 10. So now we can compare compound 5 with compound number 10. So compound 5 was the previous antibody-drug conjugate, and remember, it's not potent on Jurkat cells because they don't have CD22. And we had mostly potency against BJAP and WSU, which are lymphoma cancer cell lines. And look at that, pretty good. I didn't notice before, I guess, but yeah, they don't have a number for Jurkat here. I imagine it's dead because they're pretty much all of the antibody-drug conjugates are negative or poorly potent against the Jurkat cells. And also, just like before, they did this target-dependent study. So they made compound 11, and instead of using the CD22 antibody, they used NAPI antibody, and that was all so it's the alternate antigen, and yeah, it's totally dead also, even with the nice tubulysin propyl one. So this is just another control study. So who's our good one now? So first it was 5, now it looks like it's 10, right? That seems like maybe an improvement because the propyl ether appears to be maybe more stable. And again, in this table, 5 and 10 look kind of similar, right? But the thing is, this is an in vitro study. This is not like in an animal or something. This is just looking at cells and trying to kill cells. All right. Okay, and so now let's look at the in vivo efficacy of this second. We're calling a second generation Tubulysin ADC. Remember, in the grand scheme of antibody drug conjugates, I'm kind of grouping all the tubulysins and calling them third generation, but in this study they're calling this second generation. It's kind of like compound 5 was a first generation, compound 10 was a second generation one, right? So in vivo evaluation of the conjugate was necessary to fully understand how changing the structure could impact the ADC stability and efficacy. So thus they then evaluated the anti-tumor effects of this new CD22 antibody tubulysin propyl ether, PR, compound number 10, in a human lymphoma xenograft model. So again, they're shooting human lymphoma cells into a mouse, and then they're studying, does this drug have a good effect or not, this antibody drug conjugate. And here's the compound. And again, 10's the good one. 11 was the NAPI one, which was the alternate antibody. And that's a big change. These are the kind of the cool things they did. They're using the ammonium linked one to give them the methyl and the cleaved product, and then they're using the propyl ether here. Okay, and okay, so let's look at the graph now. And what we're going to show is the stabilized tubulysin propyl antibody drug conjugate number 10 resulted in tumor stasis for 21 days when administered at 1 mg per kg. So tumor stasis means like no tumor growth. That's a good result. It's showing it's active, right? And Yeah, so there's a lot going on here. The red one is the competing one with MMAE. And then we have 2 doses of the compound number 10, which is kind of low dose, high dose. And then on the high dose, it seems like there's less tumor growth. And then we have the vehicle out here on the left, which is like no drug. And also we have the NAPI one, which is the non-CD22 antibody. Well, look at that. It just goes right with the vehicle. So that shows that, you know, having the wrong antibody will totally prevent this stuff from working, even if you have a super cytotoxic peptide on there. Okay. And then it also appeared that their compound number 10, appears to be better than compound 8. That's kind of cool, right? Response was dose dependent with modest tumor growth at 0.5. So this is 1 mg at the bottom, and this is 0.5 mg. So that's cool. It's dose dependent, right? You want it to be dose dependent for it to appear like a real result. That looks good. That looks like compound 10 is having a good effect, right? It's also target specific. Like I said, the anti-NAPI one, compound 11, which basically follows vehicle. It's this graph is totally inactive, right? That makes sense. It's a good negative control in this study. I noticed this just today when I was preparing this PowerPoint, but is this really a fair comparison, fair head-to-head matchup? Because it actually looks like compound 8 is at 0.5 mg per kg, and the really potent one at the bottom is 1 mg per kg. So that's not exactly a head-to-head matchup. It almost looks like in a head-to-head matchup, MMAE is better because it's at 0.5 and this top line's at 0.5. So that's weird. I mean, it actually kind of makes all of this look like maybe garbage data, but however, there's a redeeming feature, which is going to be the next thing. There's more compelling data. This graph is actually a little misleading, though. I hope you can agree that it's a little misleading. Okay, yeah, some science is a little misleading, and I do believe they mentioned this in the text too. I should go read it again. Okay, so very lastly, now they're going to actually look at the multi-drug resistance. So one of the most compelling reasons to utilize tubulysin as an ADC payload versus MMAE payload is their potential to overcome multi-drug resistance, right? And we said that from the beginning, that tubulysins maybe are better at solving multi-drug resistance. So as they previously reported, I guess as Genentech previously reported, they derived a lymphoma mouse xenograft model so it's a xenograft model that's resistant to MMAEs. Okay, so they actually kind of created a special cell line that maybe has a high PGP, the P-glycoprotein efflux pump. Okay, so these models were characterized to have upregulation of PGP, the efflux transporter, right? That kind of nasty one that kicks out drugs from tumor cells, while maintaining equivalent levels of CD22. So it looks like Genentech, I think it was Genentech, they created like a mouse. They call it it's a mouse model. It's a mouse model of multi-drug resistant cancer, or maybe it was the cells that they inserted. But anyway, it's a model of multi-drug resistant cancer. I'm not exactly sure how they did that. It's some kind of cell biology magic, maybe. They used this model as it represents acquired resistance to the previous generation of ADCs targeting tubulin, right? So this new model might be a good model for to really test the good drugs to see if they work. So thus they tested tubulysin PR and MMAE head-to-head. Now they actually did it head-to-head against this BJAB Luke PGP lymphoma model. So it's a mouse model of multi-drug resistant lymphoma. This is table 4C, and this actually did show them the right result, and it is not misleading at all. So MMAE-based ADC was dosed up to 8 mg per kg. That's a large dose of these drugs, and it basically had no response. These red lines are, we'll say, minimal response. It's like not really doing very much. Whereas their new guy, compound 10, that they're excited about, gave clear dose-dependent tumor growth inhibition, resulting in 74% tumor growth inhibition at 2 mg per kg. So the triangle, which is the bottom line, that's a pretty small dose, right? And it's showing a pretty noticeable effect, certainly better than the competitor drug, compound 8. All right, so compound 10 is doing the right thing. And then, of course, compound 11, again, that's the NaPi one, which is the other antibody, not really having much of an effect at all. Maybe almost as good as compound 8, almost. So compound 10, good. The improved activity of tubulysin propyl ether antibody drug conjugate relative to MMAE in this multi-drug resistant model is likely a result of the released tubulysin PR payload, the drug, not being effluxed, right? That's what they said all along, was that tubulysins are maybe resistant to efflux. Very cool. Okay. And so now it looks like compound 10 is actually doing the right thing, certainly in this model of multi-drug resistant cancer. So conclusions of this paper. So yeah, the propyl ether looks good, and the methylammonium thing looks good in compound 10. Compound 11 is a bad compound. It's the wrong antibody or different antibody. So their conclusions are: we have employed a peptide linker combined with a quaternary ammonium salt linker connection to provide a stable and bioreversible connection of tubulysins to antibodies. So it's stable but bioreversible, and that means it can be cleaved, and the cathepsins do the cleavage and it falls apart. Okay. Furthermore, we have identified and addressed the metabolism of an acetate ester critical to activity with its replacement to a propyl ether, right? So the propyl ether, good. The resulting conjugate was highly stable and efficacious against both drug-sensitive and multi-drug resistant tumor models. I would say that against the drug-sensitive one, the MMAE one is, it's not exactly clear. Their data was not totally convincing. I mean, that this was actually better against. They're saying what they're saying is probably true, but right, but that it's a little misleading because they didn't do a head-to-head matchup in the drug-sensitive one. The multi-drug resistant one, I think there's no, which is probably the one you're more, that you care more about. It definitely did really well. The latter activity substantially differentiates this tubulysin antibody drug conjugate from the clinically validated MMAE ADC and generates excitement for its further investigation, right? I think that's pretty clear, certainly for multi-drug resistant. And another way to say that is, we want money, 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 right? So, I mean, and you know, that's what companies do. And I think that there's some pretty promising results here, maybe against multi-drug resistant tumor cells, which people get, right? And that compound 10 might be a good clinical candidate. Okay, parting thoughts, and this is actually related to the homework assignment. The homework assignment has a little bit from this last paper, but there's actually a big goal is to interpret a different paper, and it's really cool. It's going to be kind of related in some concepts, and it's not a super hard homework assignment. But first of all, what was the name of the phase 2 metabolite that looked like glucose, kind of, but it had a carboxylic acid attached? Do you remember that phase 2 metabolism? Let's call it XXXXX, okay? All right, so that, you have to think about that one. If there was an enzyme that would cleave it, what might that enzyme be called? I don't know. What do you think? Enzymes that cleave things maybe are called ACE, like peptidase and protease and stuff like that. So it might be called blah, blah, blah ACE. Wouldn't it be cool if blah, blah, blah ACE was expressed in high quantities in a tumor microenvironment, so a place where tumors are, leading to tumor-localized drug delivery. Wouldn't that be cool, right? And then the trick would be maybe we could attach this XXXX thing on our, like a drug conjugate. And it doesn't even have to be an antibody drug conjugate. It could be some other kind of drug conjugate. But if it could be cleaved and release the drug where a tumor is, then that could maybe be a cool localized drug delivery device, right? So this is going to be the paper. It's Leganan et al. JMed Chem 2012. So it's a little old. I mean, the other paper, the antibody drug conjugate one we did was 2017, so a lot newer. But this is still cool too, and it's a fun paper. I don't know. I think you'll enjoy it. You're going to have to probably read it a couple times to try to understand it. I'd probably recommend read it first before trying the homework, and then just go through the homework questions. This is one of the key figures, and it kind of shows the basic idea of the structure of this thing. Doxorubicin is a cytotoxic molecule. It's kind of like what tubulysin was in the last paper. And then they have doxorubicin also contains this sugar. So this is all part of doxorubicin, right? This thing, which kind of looks like one of the phase 2 metabolites, that can be cleaved with an enzyme, right? Something that maybe be called XXXXX ACE might cleave this. Well, and then this is a self-immolative cleavable linker, which once this gets cleaved, it kicks in, breaks off, and kicks off doxorubicin. So you'll see that in the paper. Really cool. And then the other thing is there's this crazy thing. This is a PEG. Remember PEG? Polyethylene glycol 10. This is a long linker. It's hard to draw here, so they just have parentheses and 10. This is a bunch of like oxygen, carbon, carbon, oxygen, carbon, carbon, oxygen, carbon, carbon. It's a big long linker. We saw this when we talked about hydrogels, right? The drug delivery from that company Prolinx, PEG, polyethylene glycol, right? And then they also have maleimide bearing side chain, which is this is attached to cysteine, right? Just like in the antibody, but this is not going to be using cysteine. So read the paper. It'll be really cool. The big difference is that this is going to be attached to albumin, right? Albumin is a protein that's in our blood. So you inject this into somebody's blood, and then albumin attaches via cysteine to the maleimide. And then it travels around your body. Travels, and then at the tumor site is an enzyme that cleaves the thing and releases the drug and kills the tumor cells. So that's what you're going to be. That's your homework assignment. Very cool. You got a week for it. All right. Okay, and then lastly, one tiny synthesis thing. In their synthesis, they used the deprotection of an allele-related protecting group, and I need to give you tips on that mechanism. So this is going to be the basic idea. Is an allyl ester, for example, allyl ester. Palladium 0 cleaves these, and I got to show you how to do that. And I'm going to do that with my camcorder. Okay, I think that's the end of this lecture. But I'm going to have a little, I'm going to do this last little part with my camcorder. But, oh yeah, that's what the PD0 thing. It's called tetrakis triphenylphosphine palladium. It's this thing. It's a bunch of there's a palladium in the middle, a bunch of benzenes and phosphoruses and stuff. Nucleophile, there's different nucleophiles. This is one. This thing tautomerizes, and you have a enol form. This nucleophilic, it attacks. We'll see how the nucleophile works in the mechanism. ND-MBA, dimethyl barbituric acid, another one. Tautomerizes, you got a nucleophile. The big thing is you need a nucleophile. And in the paper that you guys are going to be reading, it's aniline, so it's phenyl NH2. Okay, so let me show you the mechanism, and then that'll be all done.